Hi, in this video, I will talk about how to trace a cell secretory pathway. So we know that for cell, proteins are produced in the endoplasmic reticulum on the ribosomes, and then they are packaged into the Golgi, and from Golgi they move to the membrane. If it's a membrane bound protein, then it would be it would be in the membrane bound condition, and if it's a secretory protein, then it would be secreted outside of the cell. Now the question is how to prove that this is the secretory pathway of the cell, and the secretory pathway of the cell includes Golgi, and then to the plasma membrane. In order to do so, we can perform one experiment. So we know that VSVG, which is actually vesicular stomatitis virus glycoprotein, which is a membrane protein. So it's actually displayed on the membrane in case of virus infected cells. So what we can do, we can take this gene sequence for VSVG and we can tag it with the N-terminal GFP, and we can clone the whole thing in a mammalian expression vector. Then we can transfect it into a mammalian cell culture. After transfecting it into the mammalian cell culture, what would happen? The VSVG containing expression vector would be expressing into the nucleus, and inside the nucleus, from this expression vector, mRNA would be produced, and that mRNA would go outside the nucleus and it would go to the ER. There the translation would happen on the ribosomes, and it would follow the cell secretory path. Now if it is following the cell secretory path like ER, then Golgi, then plasma membrane, then over time, if we record it in a live cell imaging, that we would find that in several time frames, several components of the cells are labeled. So for instance, initially ER would be labeled, then we would expect Golgi would be labeled, and at the end plasma membrane would be labeled. So several researchers did same kind of experiment and found that in the 0 minute you can see the ER, which is around the nucleus, is labeled, and then at 40 minutes time after starting the recording, you can see the Golgi is actually marked by GFP. That means after 40 minutes the whole protein is actually moving to Golgi and actually lighting up the Golgi. And if we wait 100 minutes, then what would happen? This whole plasma membrane of the cell would be actually illuminated. So that also suggests that our cell secretory pathway is from ER to Golgi to plasma membrane. So that concludes the video on the tracing of cell secretory pathway. If you like my video, give it a quick thumbs up, and don't forget to subscribe, and please leave your comment below. Thank you.